Hi, everybody. Welcome to this webinar on developing tools for climate-smart coral reef management in the U.S. Virgin Islands. If you're just coming in, you can put your name, where you're from, and your position, your work, in the chat. It'd be great to see who you are and get to know you a little bit. So this webinar is brought to you by the Nature Conservancy Reef Resilience Network in partnership with the NOAA Coral Reef Conservation Program. My name is Annie Cross, and I am the lead scientist for the Reef Resilience Network. Today, we will be joined by the team from the Nature Conservancy in the U.S. Virgin Islands as they dive into their coral program and guide us through the work happening in the U.S. at the USVI Restoration Hub. They will show how they're building restoration approaches that can succeed in the face of climate change. From genetics to drone-based mapping of resilient coral colonies, the team will walk us through the strategy they're developing to meet the challenges of a warming ocean. So this webinar will last an hour with a Q&A question and answer session just after the presentation. We're recording this webinar, and so the recording will be available in the next few days after on the YouTube channel and on the Reef Resilience Network website, and we will also email you the link. So for those who don't know us, I wanted to do a quick introduction to the Reef Resilience Network. For the past 21 years, the Nature Conservancy Reef Resilience Network has helped marine managers and practitioners around the world effectively manage, protect, and restore critical marine habitats. With the help of more than 400 global experts, we connect marine managers with the latest science and conservation tools for coral reef management. We connect managers with experts globally, and we provide training and mentorship to support managers and coral reef conservation and restoration practitioners in their work. We've trained over 61,000 people over the last 21 years from more than 94% of reef location. You will find most of our resources on our website, reefresilience.org, including our free online courses, the self-paced and the mentored ones, toolkits on topics critical to effective management, case studies from around the world, summaries of new journal publication from our open access library, and webinars, recording of webinars such as these, and our podcast. So now I'll present our three speakers. Jessica Ward, who's the Virgin Island Program Director, will be followed by Catilin Lustic, the USVI Coral Program Manager, and Dr. Steve Schill, the lead scientist for the Caribbean Division, and actually Director of Science and Strategy now for the Caribbean Division. So I will pass the mic to Jessica for this presentation. Thanks.
All right. Good day, everyone. Thanks for joining us. So the big picture of what we want to present today is what are some of the methods that can be used to build climate resilience into coral restoration programs. So Caitlin and I will first talk about our restoration program on St. Croix, and Steve will follow up with a presentation on the use of drones for reef habitat mapping and how that information can be used by restoration managers for decision making. So those of us in this field know it's been a rough few decades for coral, with the threats and decline continuing to increase. Coral reefs have suffered greatly over the last 50 plus years, especially in the Caribbean where we live, where live coral cover has declined 50 to 80% since the 1970s. We've had disease that has all but wiped out elkhorn and staghorn corals, previously the two most abundant species in the Caribbean. And other diseases have since appeared affecting different species. We have chronic problems like water quality degradation, sedimentation, overfishing, destructive fishing practices. And then, of course, in 2014, a novel disease, stony coral tissue loss disease, appeared in South Florida and then spread to most of the Caribbean over the next few years. This disease differs from previous coral diseases that it's easily spread, highly virulent, and affects over 20 species of coral. And we now know that the biggest threat to coral reefs today is climate change. This is driving impacts like increased frequency and severity of storms, sea level rise, ocean acidification, and of course, increasing global temperatures and heat waves. So this can also happen in the ocean. And while marine heat waves and periodic coral bleaching are a natural phenomenon, these events, like storms, are increasing in frequency and severity. Bleaching is a process by which corals eject their symbiotic algae as a stress response. These algae give them their colors, they provide them with the main source of energy through photosynthetic products. And if coral is bleached, that doesn't necessarily mean it's going to die, right? They can recover if the heating abates in time. But the problem comes when it's really prolonged. If a coral is severely bleached for too long, it can be weakened, it can become more susceptible to disease, and eventually mortality can set in through starvation. So in response to these threats, the Nature Conservancy began conducting coral restoration in the Virgin Islands in 2009, but was largely focused on acroprid corals, particularly Acropora cervicornis or staghorn coral, because it's easy to handle underwater without the need for a land-based facility. In 2020, we received funding to construct a land-based nursery that allowed us to work with slower growing boulder corals using microfragmentation methods to speed growth and propagation and scale our work to cover greater areas of reef than we had been able to do so previously. The Coral Innovation Hub on St. Croix came online in around 2021. It's when we really got most of our tanks filled and really start to get to the point where we can start putting corals in there. So the hub is an ex situ coral nursery comprised of 6 upper and 18 lower raceways. We have 2 different systems, technically multiple systems, but there's 2 different groupings. We've had our 6 tank system for a bit longer than our lower system. Our lower system was expanded with all this additional funding that we've received to scale our work. And so these systems now contain state-of-the-art automated life support systems. We have a dry lab. We have a wet lab to support experimental investigations in larval recruitment and rearing. Continued modifications, improvements, additions to the infrastructure are ongoing to continually improve how we grow corals. A seawater intake pipe was installed in 2022 that allowed us to move to a semi-open system with higher flow and turnover. And funding from the local government in 2023 allowed us to make several additional improvements, including purchasing redundant emergency generators. We live in a hurricane zone, and any of you that live in small island nations know that we are prone to very frequent power outages, which can be problematic for these types of systems. We installed a 25,000-gallon cistern with chillers that provides a reliable source of fresh, clean seawater during times of impact offshore. So if you have really hot water conditions and you want to retain this sort of like a stash of water, if you will, when you don't want to bring in that water from offshore for all kinds of reasons. We've all been inundated heavily with sargassum over the past few seasons that can really impact our water quality that we're intaking. So these types of sort of backups and redundancies can provide continued ideal conditions for these corals throughout environmental impacting conditions. We also have a new rigid structure over our lower raceway that protects the corals from excessive UV. We can shade our corals a bit more. And we also protect from things like heavy rains. It can reduce the impacts of heavy salinity swings. Next. So the early funding the hub received also funded our first large-scale restoration endeavor across roughly 50 acres of reef within the St. Croix East End Marine Park. This seemed a little ambitious and frankly daunting task in the beginning, especially since we had a very small team. Additional funding allowed us to grow our team, and brainstorming led to the idea of these sort of mass outplanting events or blitzes, where we chartered a large vessel and brought a lot of volunteers, especially from partners in the region, to add to our staff and create these large teams that could go out and create essentially these underwater assembly lines and put a live coral down in a very small amount of time. This method has allowed us to scale up production to the point where we can outplant up to 10,000 corals annually with a field team of less than 10 people. So we've been very successful, I guess you'd say, operationally with scaling our restoration work. Next. But then in 2023 and 2024, which saw the most severe marine heat wave ever recorded, resulted in a mass coral bleaching event that unfolded globally with unprecedented numbers of degree heating weeks. So the U.S. National Oceanographic and Atmospheric Administration, or NOAA, developed the Coral Reef Watch to aid resource managers in predicting when bleaching would occur. Previous mass bleaching events have typically seen around 10-degree heating weeks, which is equivalent to an alert level 2, or risk of reef-wide bleaching with mortality of sensitive corals. But then in 2023, many areas saw more than 20-degree heating weeks. Because it's so severe and so unprecedented, NOAA actually added alert levels three through 5, which are defined by heightened risks of mass mortality. So our monitored reefs experienced more than 90% coral bleaching in those years here on St. Croix. And some species, such as the acroporids, experienced roughly 90% mortality. Those numbers are just kind of a guesstimate right now. They might even be higher. We're still processing that data. So even though operationally we're able to scale our outplanting methods and get a lot of coral out there in a short amount of time, like everyone else in this field, we now have to pivot on focusing how to build resilience into the scaled production so that those corals have the best chances of survival through these ever-increasing impacting events. This forced our program to rethink our strategy and start thinking more concretely about incorporating methods that help select for or build thermal resilience. We've been assessing various techniques and methods to develop a toolbox for integrating thermal resilience into our work using both spatial and organismal metrics. So I'm going to pass it on to Catilin, who will talk briefly about how we're incorporating each of these techniques into our restoration portfolio. But we want to stress that most of the work is still ongoing and we don't necessarily have all the answers yet on what works.
Thanks, Jessica. So as Jessica mentioned, the fourth global bleaching event in the summers of 2023 and 2024 caused widespread bleaching and mortality in the U.S. Virgin Islands, particularly in our elkhorn populations. By using in-water structure for motion photomosaics and drone monitoring, the team has been able to identify both areas that could serve as refugia, where the impacts were less severe, and individuals that survived either because of their location or genetics. Steve's going to talk more about the drone monitoring in his presentation, but the in-water mosaics allowed us to assess impacts and resilience at a finer scale across a wider range of species and at sites deeper than the drone allowed. Using this information, we can start to think about future areas where elkhorn restoration might be more successful, and we've already begun sampling from the individuals that survived to be represented in our land-based gene bank and to continue to propagate for future restoration efforts. We've collected from about 200 elkhorn colonies around St. Croix at 10 different sites, and we have maintained them in the lab as assumed genotypes. These represent all of the genotypes in St. Croix that we know are still alive following those bleaching events. We recently took genetic samples and are in the process of analyzing the results to understand how many true independent genotypes we have represented in our lab. And this will allow us to ensure that we're mixing genetically distinct individuals at outplant sites and plan for upcoming spawning work with Acropora palmata. We have not fully analyzed the results yet, but an initial look suggests that the remaining genetic diversity is low, even across different sites. And if this is truly the result, we'll need to think further about how we best support recovery of this species. We're currently focused on genetics for Acropora palmata because we know that the genetic diversity was already limited and then further declined in 2024. But we're also interested in conducting genetics on the other species being held at the lab and in better understanding the symbiotic communities. One way we might think about supporting increased genetic diversity is through assisted gene flow. Assisted gene flow is the managed movement of individuals or sperm and eggs between populations within a species range to mitigate local loss of genetic diversity and the potential for adaptation. We're making plans now to accept elkhorn corals that are currently being housed at Mote Marine Lab in Florida that are crosses between parents from Puerto Rico and Curacao. We've worked closely with the Department of Planning and Natural Resources here in the USVI to develop a set of conditions that minimize risk to local populations. So this involves a series of steps and assessments before any assisted gene flow corals can be outplanted to our local reefs. We're also collaborating with Sea Corps and other Caribbean practitioners to lay the groundwork for the future exchange of larvae. This collaboration would help distribute genetic diversity across the entire region, particularly for species where environmental stressors have caused sharp declines in their populations, resulting in limited genetic diversity. So in the lab, we're able to conduct thermal testing to determine relative heat tolerance among our stock. particularly for Acropora palmata, because we know they're highly sensitive to heat stress. We propose to build ED50 models for the different genotypes. This model quantifies the temperature at which a coral colony experiences a 50% decline in photosynthetic efficiency or chlorophyll content. And in simpler terms, this is how effectively the coral can turn sunlight into energy using its symbionts. This analysis can provide context around differential responses across genetic individuals. And this information combined with the genetic testing that we're doing can help us tease out whether the results we're seeing in the field are due to environmental factors or genetics. If we find that our genetic stock is highly related, then this model might be less useful, and we might need to think instead about areas of the reef that provide refuge from the more extreme temperatures. In conducting thermal resilience testing, we plan to use multiple methods, including relatively short-term testing, similar to the CBAS methodology, but also longer-term temperature experiments that might more accurately mimic the ramp-up of temperatures on the reef. All of these pieces of information put together can help us make informed decisions about how to support recovery of the species. In the past, we conducted sexual reproduction and grow out in field-based nurseries. In anticipation of bleaching in 2024, a subset of recruits that were settled in May 2023 were brought into the land-based nursery for safekeeping. And after about a year, we noticed a significant increase in growth as compared with the corals left in the field. A portion of each of the land and field reared corals were then outplanted, and they're being monitored over time to see if that increased growth rate in the lab translates to higher growth rates and or resilience to bleaching in the field, and how long that might last once they're outplanted. Understanding how this early life stage boost translates to long-term outcomes can help us determine whether land-based rearing of recruits for the first 6 months to a year is worth investing in each year. We also have been settling larvae in water that's at about 29 degrees Celsius and then rearing them in warmer water as well. The corals in the worm systems have had higher survivorship rates and are larger than the recruits in the systems that are set at ambient temperatures of about 20 to 26 degrees Celsius. But the recruits in the warmer system have fewer symbionts. Subsets of these recruits have been outplanted and distributed throughout the lab to see how they continue to grow and survive. We don't have long-term results yet of this study, but we are hoping to see higher bleaching resistance among the coral settled and raised in warmer temperatures. Another method we plan to employ this summer is to create spawning hubs for Acropora palmata. The mortality we've observed has drastically reduced total reproductive output. So we plan to implement facilitated sexual reproduction for the first time with this species. Sexual reproduction with Acropora palmata is particularly challenging. The spawning occurs over a 15-day window. They prefer high energy shallow reefs that can be difficult to navigate. And the significant decline in the population means that colonies are more dispersed than they once were. So to establish genetically diverse in situ spawning hubs, we'll place large fragments of each genet at one or more strategically placed sites chosen based on the information we've gathered about where this species seems to be still be doing relatively well. By concentrating a diverse cohort of corals into a small area, we can safely monitor many unique genets with fewer divers, vastly reducing cost and effort required. In addition, we hope to enhance natural reproduction by increasing the likelihood of cross-fertilization between previously geographically distinct corals, and the planted corals will grow and fragment at those sites and help repopulate those areas. And then finally, we're conducting a large-scale spatial analysis of reef areas in partnership with our Caribbean Division science team using remote sensing imagery to assess areas of coral loss versus possible refugia. And I'm going to turn it over to Steve now to talk more about that work.
Hey, thanks, Caitlin and Jessica. Yeah, this has been difficult these last few years, seeing the dramatic mortality rates across these reefs, and it's a all hands on deck moment, and the work that the Coral Innovation Hub is doing is really cutting edge, and it's exciting to be part of it. I'm going to present some of the work we're doing in terms of monitoring these reefs using drones. I want to acknowledge Dr. George Raber. He's been critical in all of this, and also Matt Davies has helped with a lot of the analysis. We know, next slide, we know that reefs, they're critical to monitor, and we can monitor them at different scales. And Caitlin was talking about in-water mosaics. That's important. We can gather information at that scale. Greg Asner at ASU recently published a paper in collaboration with Planet on the ability to detect live coral from space using the Tanager satellite, and that's another big benchmark in remote sensing to be able to monitor that. And drones in the last 10 years have really filled a niche in terms of being able to map at the colony level across kilometers of reef. And so this is something that we've been doing since May 2021, is monitoring these sites across USVI and trying to answer the questions of, can we map at the colony level, and can we map the impacts of these two very strong bleaching events, and then identify where the remaining colonies are. And so we're going to walk through that a little bit. Next slide. So we're trying to develop information for managers, answering questions on where the coral are, how much do we have, and how different across space are these colonies in terms of their live coral occurrence and also the bleaching. We want to understand how extensive the bleaching is across the reefscape. We want to understand these spatial patterns. And one thing that really has been helpful is using drones to identify remaining colonies, and then using that information to help inform what Caitlin and Jessica has just talked to us about. So we really want to use this information to prioritize management decisions on where we can intervene and where we need to do additional monitoring. Next. So back in 2014, we started dabbling in drones, and we developed our first drone. It was a 3D printed hull with electronics in a Tupperware strapped onto the hull, and a GoPro. I think it was probably a version 3 GoPro. And our desire to develop this was to do field surveys in areas where we couldn't get the boat because it was too shallow. And so this was our very first model that we developed. And then we continued to adopt new technology. This technology changes very rapidly. We use 3DR solos with a full frame QX1 camera, modified GoPros strapped to the bottom. We used those for a few years. And the early days of drones, you had very limited time in the air. You had maybe 15 minutes. That was a good flight. The cameras weren't as good. And over time, these drones have developed further and further. So we're getting better cameras, we're getting better flight time. And the most recent drone we're using now is a Wingtra Gen 2 drone that we purchased in January 2023. And with this camera, we have a 61 megapixel camera that we're able to fly at 200 feet. We're able to fly about 50 hectares on a battery. It flies for about 40 to 50 minutes. And then at 400 feet, we can map about 500 hectare on one battery. So the ability to map has increased substantially over the years. Next. So what we're going to talk about is the Acropora palmata species. With drones, we're able to map in clear water conditions down to about 3 meters before there's too much refraction in the water. But this is a species that occurs in those reef building areas, shallow, high energy. So we're going to talk about some of the the experimentation we did to be able to come up with a solution to map these across a large reefscape. Next. So those of you that have mapped using drones, you know you can develop a flight plan. You identify several parameters, the overlap. Uh, you have to make sure that you identify the type of camera you're using. And once you do your mission planning, you execute your mission. You have to specify your flying height. What are your resolution requirements? Because what you're trying to map is based on that flying height, and you need to determine at what level you can discern and detect the features you're trying to map. And these images, these stereo images that are collected, are then processed to create this ortho image that you can analyze. And we use several different softwares and algorithms to process these imagery, and each of them have their pros and cons. Next. So after testing, we determined that flying at about a 65 to 75 meter altitude enabled us to get the resolution we needed using the camera we had to be able to map at the colony level. The higher you fly, the more area you can map. And so we're trying to maximize the area, but at the same time, map the features that we're interested in. Next. Now, there are many things that you need to consider when you're flying over water. If you're flying when the sun is high, you get what are called caustics and patterns in the water column that and sun glint, things like that, because the angle of the sun is high and it's reflecting back into your sensor. And so you need to plan your missions accordingly. So low sun angle, either in the early morning or later in the day, are the best to acquire imagery over water. Next. There's also environmental conditions you need to think about. Following rain events or storm events, there can be turbidity in the water column, which prevents you to see through and detect what you're trying to map. During stormy days, there can be a lot of wash with the waves, a lot of sea foam and energy. So ideally, you want to fly when it's calm conditions, and also sargassum, if there's things that are floating on the water surface. You want to try to minimize that. So a lot of planning, and the stars really need to align when you're trying to get the best imagery possible. Next. Also, it's important to make sure you're capturing your data at a fast shutter speed. The lower you fly, the more susceptible the imagery is to image blur. So you need to really employ a fast shutter speed. Getting a message here on my Zoom.
No, you're good, Steve.
Okay,
We're still going. Yep.
okay, good. Okay, go to the next slide. So make sure that your shutter speed is fast so you can capture those features in crisp detail. Now, also, when you get good imagery, you also need to get field data to supplement and validate that imagery. We have been using a drop cam survey, but this is not really practical in shallow areas. And so go to the next slide. What we've developed is a floating platform. We attach our camera to a GNSS receiver in a Pelican case. That Pelican case is waterproof. We have a base station that we post-process kinematic correct those coordinates, because when you're flying with drones, you want to make sure that those pixels are aligned to their geometric position as accurately as possible. And so we're both geometrically correcting the imagery, and we're geometrically correcting the photos that we're taking. So every time we click that shutter, it's stamping a coordinate, and that coordinate is post-processed to as true a position as possible. It's typically 2 to 3 centimeters based on the base station. So this is very important when you're trying to validate your imagery. Next. So one of the first studies we did was working with Lisa Karn at Fragments of Hope. We evaluated the impact of restoration and the increase in different species that they're restoring. This is back in 2019 and 2020, and there's a paper that talks about that work. Next slide. And we also were able to join our California chapter and work with coral gardeners to do drone monitoring in Moorea. And this was to help coral gardeners identify areas to outplant. Next slide. Here's some imagery from that mission. You can see the coral nursery sites there on the left. Go to the next slide. And it was really phenomenal conditions because these corals are within the lagoon, very calm water conditions. We could get really good detail, and the variety of coral that was there was impressive. And we used e-cognition. Go to the next slide. First, we worked with the locals, coral experts, to identify the different species within the drone imagery. You can see some examples here. This was flying at 65 meters, and we were using a 42 megapixel camera. Next slide. And then take this imagery, which was acquired at a 1 centimeter pixel, and then run it through e-cognition to be able to map. Next slide. The different species that we were seeing, and they were particularly interested in the algae that was growing on some of these coral colonies, and identifying across 1/3 of the island. We were able to map these different species. So this shows you some examples. Let's now move on to St. Croix, and we're going to show you what these data sets look like. Here we are zoomed in very closely on a data set that was acquired in August of 2023. And if you keep clicking through the slides, you can see how many coral colonies there were. And we consider this, the maximum coral cover for this area, which is about a 7-kilometer stretch. Yeah, keep clicking, and you can see what a large area this is, and how drones really fit this niche of monitoring at the kilometer scale, because having divers in the water to be able to assess this would be very difficult. And using satellite imagery, you're not able to detect at the colony level unless these colonies are very substantial. So we've been monitoring these areas since 2021. We've been trying to time it over ideal conditions. Sometimes it's successful. Sometimes we have stormy areas. We have sargassum in the water. Typically, areas of between August and October, we can have very calm days, which is a good time to acquire the imagery. Next slide. And let's zoom in and show you some examples. When you're doing deep learning, which is what we're doing here, you need to be able to train the deep learning algorithm, and we use image segmentation to be able to grab those segments and use them for training. Here you can see the segmentation algorithm that's able to recognize and grab these colonies. Next slide. And across a 2-kilometer area, for example, in this particular reefscape called Lew's Reef, we're able to apply the deep learning detectors. You have to train. We're using ArcGIS deep learning to be able to train across an area what you're trying to identify. Deep learning works very well when you've got a feature that has a contrasting background, and that live coral color of the Acropora palmata stands out, and we're trying to develop training data that is consistent and identifies the different patterns that these corals can have. And across about 2 kilometers, we've mapped about 3,100 square meters of live coral. That was in August of 2023. And so we can use that as a baseline to see the loss of coral over time. Next slide. We also use it for identifying coral bleaching. You can see a time series here. As that bleaching event occurs, we can use drones to detect what colonies are bleached and come up with an area number. Next slide. We can look at patterns and trends over time, which colonies survive bleaching events and which colonies die. And we can map that, we can quantify that across time in June 2023, comparing our live coral in Acropora palmata to what we mapped in March of 2024. Next slide. And just last year in June, we were able to collect new imagery to identify the remaining colonies. And this has been very helpful for the genetic work that Jessica and Caitlin are doing. Snorkeling across this and validating it with in-water photos, being able to fly and identify where there's potential remaining colonies. Next slide. So here's an area in St. Croix where we hadn't flown before, but we flew it in May of 2025, and we were able to identify colonies that had survived the 2 bleaching events. And so this is a great use of drones, is in being able to find these remaining colonies across a large area. Next slide. There are limitations, like I mentioned. Identifying colonies that are deeper than 4 meters, it can be difficult unless you have ideal conditions. And also there's a certain size threshold and you can adjust that based on your flying height, but really small colonies can be difficult to detect in the deep learning algorithm. Next slide. And also, when a Acropora palmata colony dies, it can be encrusted with algae. And so that can have a very similar color. And so that's something that we need to train on to be able to recognize the difference between these false positives and what a live colony looks like based on a dead colony that has encrusted algae. Next slide. And real quick, I want to tell you another example. We've been working with FAU, with Megan Davis, to use this type of approach to map queen conch in shallow seagrass beds. Next slide. Here's some examples of what that looks like. We've been looking at the difference in processing algorithms, what we're able to get with desktop processing of the ortho imagery versus cloud-based, using different platforms such as the Mavic and the Wingtra and the altitudes, what kind of imagery we get. And we can also detect movement of conch over time. So that's by flying at 2 different times, we can track individuals and see how far they move. Next slide. Here's some results of identifying the conch across a fairly large area in Grand Bahama. Next slide. And finally, I want to give a shout out to those of you that are working in low budget environments. There are a lot of tools out there. You can buy a less than $1,000 drone and use these flight planning softwares and platforms to create automated missions. There's cloud-based processing that's relatively inexpensive to do your orthomosaicing. And then there's tools like QGIS that allow you to do the analysis for free. So there are options out there for low budget drone monitoring that are exciting to see, and people are starting to adopt this using these tools. Next. So this has a lot of implications for managers. We're moving away from point surveys to looking at larger reefscapes. We're able to identify these bleaching hotspots and improve our intervention efforts. We're able to have a more rapid response. We're able to track recovery, mortality, and long-term trends, and overall just support adaptive climate-informed management by identifying these refugia areas. And all that is an example of how we can use new tools such as drones to be able to better manage these corals that we all care about. And with that, I think we're going to wrap up. Next slide. I'd like to acknowledge our local partners and turn it back to Anik.
Thanks a lot, Steve. So now I'm inviting all the speakers to turn on their camera. We're going to have a question and answer session. I see some of you have already put our Q&A tab to good use, so please continue to post your questions there. And I'm going to start with a question for you, Steve. You can turn your
Sure.
camera back on. Actually, there's a really good question that came up here. Are the train algorithms site or region specific? So, for example, can you take a model that you trained in the USVI and apply it either to the rest of the Caribbean or Florida or somewhere else without further training?
Well, we're actually, it's difficult to take a training set that was acquired in 2025 and apply it to the same location in 2025, 26, each data collect can have different conditions. As we saw, there's a lot of things that influence what the drone imagery looks like. And the deep learning algorithm is being trained on that specific data set. But the what we're experimenting with is we're stacking these data sets together. One of the things that's difficult is these are very large data sets, hundreds of gigabytes, and trying to develop a library that can be consistently applied across time to the same area is where we're trying to reach. Now, taking that and applying it somewhere else in some geographic location, that would be amazing. But right now we're developing libraries that we can use consistently in one particular location. So that's a great question. That would be the holy grail to be able to do that and share it and allow people. I think that's possible, but we're not there yet.
Thanks, Steve. I have a question for Jessica and Caitlin, and it's a little bit about your setting your goals. So the question is, what are the key performance indicators of a successful coral conservation, and I suppose restoration as well? And there's two parts to this question. So I'll start with that. So what do you look at when you're looking to know if you are being successful or not, both in conservation and restoration?
So I'll take a first stab at that. It is kind of project dependent and it can be specific to the funder. They might have specific requirements that you have to meet after a certain period of time. I think kind of generally the rule of thumb is you want to have around greater than 80% survival after a year is for our projects considered pretty good survivorship, and we have had that in a lot of cases. When we were working with the Crapper palmata, the elkhorn coral, which was the species we primarily worked with the first couple of years of our current program. And prior to the bleaching events of 2023 and 2024, we had greater than 80% survivorship and sometimes even higher on certain reef areas. So it depends on the species, the method, and exactly where they are being planted. But as far as like what we're looking for, it's really project by project specific.
Did you want to add anything?
Sorry, I had put a question out to one of my project managers. So she was saying that we had 95 to 97% survivorship in the first 6 months of our most recent outplanting at Buck Island. So really successful there.
Is that unprecedented, that level of survivorship?
I would say not necessarily. We were working with some boulder coral species, whereas we worked with primarily palmata in the earlier years. But I will also say that these particular corals this year are ones that we did condition in our lab for quite a long time before we put them on the reef so that they would have higher rates of survivorship. And we have, like I said, we haven't really dug into a lot of that data just yet, but the early findings are showing that when we are acclimating things in our lab for at least a few months before we put them out the reef, they seem to have much higher success.
And I suppose the real test is the next time you have a bleaching event to see what happens to those colonies, right? So,
Exactly.
okay. Well, to be
Not that we're
followed
hoping for
then.
that, but
No.
but preparing for it, right? Prepare for the worst.
Um, Caitlin, I don't know if you're going to be able to answer the second part of the question. It's quite quick, but do you know if Ocean Biodiversity Information System, or OBIS, is the best reference for coral species or most complete coral database globally? We don't know, do we?
No, I don't. Sorry.
Yeah. All right. Well, for whoever asked the question, sorry, we don't know that part. Um, okay, moving, Steve, we've had several questions. I know you mentioned this during your talk, but I think people are still wondering, what is the maximum depth that a drone can map? So I know you said it depended on weather conditions, etc., but rule of thumb is you wouldn't try to work with drones at around what depth? Below
Well,
what depth?
the features become more and more fuzzy. The detail that we're able to see, and that's just the inherent physics of water in a water column. So if you have very clear conditions, I mean, there's new techniques. Well, they're not new. They've been developed over time at the University of Miami, looking at taking rapid uh, photos across time and synthesizing that information to be able to really see through the water column. But we can't do that with the drones that we have. And so typically the threshold's probably around 2 to 3 meters. Beyond that, it becomes too difficult to really map. And it depends on what you're trying to map. Like if you're trying to map seagrass, which is dark, you may be able to see that at deeper depths. With satellite imagery, we can map deeper depths, but as you go deeper, the reflectance that you're able to record becomes less and less. Those light waves are being absorbed, and so it's really a shallow monitoring approach, and especially when you've got turbidity, you're going to have a really hard time seeing through the water. So
But beyond 3 meters, you have to have more specialized tools and a bigger capacity than if you're just buying a drone and trying to do things sort of low budget, I'm gonna guess.
yeah, yeah, it all comes
Okay.
down to what you're trying to map, but we're limited with how high we can fly drones, and so I would say these tools that we're talking about are really suited for features that are less than 2 meters in the water column.
Okay. Thanks, Steve.
Yeah,
I'm gonna go back to Jessica and Caitlin. It's more of a Caitlin question because I know you've been working on this, Caitlin, but so one person asked, what plans do you have for us managers in Africa, since there has been a lot of destruction of the colonies out there? Thank you. So I'll guide you with that question. I imagine that there may not be the same resources for some project. I think there's some big projects in Africa, but for some projects in Africa to build a coral hub and do genetics and get drone mapping to figure out where are the surviving, more resilient coral colonies, etc. So with lower resources, more simple setups, what can you do to integrate climate change into your restoration or conservation efforts?
Yeah, thanks for that question. I mean, a lot of this can be done just using people in the water making observations. So, during a bleaching event, spend time in the water, see If there are specific areas where you're seeing less bleaching, that might be an area where you might want to focus restoration in the future. It might be something about that reef where you're getting higher flow, or some characteristic about that area that's good for corals. But then also you can be monitoring both your corals, and if you already have corals in a field-based nursery, or if you want to collect new corals, look for the ones that are not bleaching during a bleaching event, or ones that recover afterwards. And those, we presume, have some level of higher resilience compared to others. So you can build your stock using individuals that seem to have some resilience to these conditions.
Thanks, Caitlin. Okay, back to Steve. We have a question about AI. And okay, I'll do the AI one. There'll be one more for you after, Steve. But are there any efforts to incorporate AI beyond the machine learning that you went through, Steve, to integrate AI into coral reef monitoring, detecting? You're on mute. You're on mute.
Thank you. Yeah, we're seeing AI progress very rapidly, and and beyond the what we're talking about, detection of features using AI. There's lots of different methods that you can apply to that. So the question is about how do we integrate more uh, AI technology and
More
information?
AI.
Yeah,
Yes.
AI into that. So, I mean, there's we just keeping up on what we can. I mean, we're being able to synthesize large amounts of data. Right now, I don't know what the total amount of drone data that we have, but we're just barely reaching the tip of the iceberg in terms of being able to use the the information that's in there. And this is a problem that conservation managers are having across the world is we've got a lot of data. How do we get information out of it? And so I think over time we're going to be able to get more and more information, uh, and our processing power is going to get better. These data centers that are essentially there to process information, the ability to extract and detect patterns in the drone imagery are going to improve. And so we need to invest in that. I'm sure there's things that we're going to be talking about in a couple of years that we didn't even think about. So yeah, that's all the advice that I give everyone is, you know, adapt to what is happening now, because it's going to be changing fast and we've got to keep learning and we've got to adopt, because it's going to help us in our conservation work, and it's moving fast. So we got to
It's moving
try to
fast.
we got to keep up. But that's the best way I can answer that question.
Great. Thanks, Steve. Okay, I have a question about process in your work. Following identification of bleaching hotspots or coral surviving stress events in your drone survey, how have you used this information to inform management and research? So once Steve brings that information to you, Jessica and Caitlin, what do you do with that information in terms of like, now you know where those surviving coral colonies are, what next?
And I put the implications in the chat there. Some of the things that
Oh, thank you.
I think are relevant to this discussion, and I don't know if Caitlin and Jessica want to add to that.
Sorry, if I can find my mute button. Um, you really have to look at across all of your data sets and kind of look at the big picture, which is what we're trying to do right now, right? You have to look at genetics, you have to look at larger site data, which is what Steve is providing. So in some instances, maybe it's the actual coral itself that might have some kind of resiliency in it, when we're finding like single corals in certain areas that have survived these impacting events, or maybe we might find clusters, and maybe the genetics and the typing of the zooxanthellae that they held within them show that there's no difference between those corals. So maybe what we're looking at isn't colony level resilience, but maybe it's actually the reef environment that they're living in. And maybe that's a particular reef spot that we need to focus on outplanting it because that has some kind of environmental conditioning that offers some sort of protection from whether it be irradiance, UV light, or oceanic conditions that keep temperatures a little bit lower than the surrounding reefs. So that's kind of like what you have to look at. What's all the different data telling you? And is it more spatial, or is it more at the colony individual level that we need to look at? So you kind of have to puzzle it together to figure out how to use that data. I don't know if Catilin wants to add to that at all.
But the intent is either protect further if it's an area that is potentially a refugia for thermal stress or some level of thermal stress. Use the colonies maybe for sexual reproduction in restoration. And then we don't know because we're still trying to figure this thing out. Is that correct? So looking at the big picture and what it means. Okay.
Exactly.
Great. Okay, we have a bunch of questions coming and just time for, I think, a couple more. And maybe while somebody answers, you can look at what you can answer directly. But Steve, actually, I have Pietro Scanziani. You've got your hand raised. I don't know if I can take you off mute. No, okay. Must have been a mistake. Okay, so I will look at this. Steve, from your experience, do you think action cameras such as GoPro or DJI Osmo as photogrammetry tech are reliable in monitoring and mapping habitat, especially coral reefs in environments such as the Red Sea, as drones are not accessible or allowed there?
Yeah, so any of those action cameras in water, I mean, a very simple monitoring approach would be go to the same area and take a GoPro picture every month and see how it's changing. That's remote sensing. We're taking a photo. We can do qualitative assessments, but drones are, as I showed you at the beginning, we started out by strapping a GoPro to a drone and setting it on an automatic shutter release. And so you've got to be creative in your ability to use technology. We have better cameras, better resolution, using GoPros to go out and monitor at the plot level. That answers specific questions. I wouldn't say that it's cost prohibitive to get into drone. You can set up and do drone monitoring with less than $1,000 budget by buying one of these. You're not going to get the examples that I showed you, but you're able to get imagery that you're able to analyze over time with a DJI Mini or a comparable drone where you can purchase that under $1,000 and use the online tools that are fairly affordable, $24 a month, to process that imagery, to set up your mission planning. And there's a lot of resources out there that train you how to do that on YouTube and others sites. So I encourage everyone that's interested in doing that to look into that and learn more about it on those sites that I listed on the presentation.
Thanks a lot, Steve. Well, we're at time. I did want to tell everybody about a webinar that is, so first of all, thank you all for your great questions, and thanks to the panel for your great answers. We didn't get to everything, but we tried to, and they're still typing. So, but before we log off, I wanted to tell you about an upcoming webinar also focused on the Caribbean and coral restoration, advancing coral restoration together in sites from the Dominican Republic Marine Innovation Hub. That's going to happen March 19th at 11 a.m. AST. So that's happening. It's from our colleagues. If you're interested in this region, go to that. And then finally, I just wanted to, next slide, please. Thank you so much. And to bring you to our site, if you have any question about any of these, I saw questions about coral reef and algae and things like that. We have a lot of resources on our website for beginner coral reef managers or people just getting into coral reef conservation, all the way to expert managers who've been in the field for 30 years. You will find things that are of interest to you there. So I really hope you go there and look for what you need. That's it. Thank you very much again, and we'll see you on the next webinar. Thanks.